Okay, so our last section, I don't want to leave it on cancers. Let's talk about this genetic manipulation. The last bit. This is it's not. It's new and it's not new because we've been manipulating and genetically manipulating organisms for a very long time before we even knew about DNA. We've been selectively breeding plants and fruits for the purposes of farming and animals. We've genetically manipulated wolves into being dogs. We've genetically manipulated the auroch into being cows by selective breeding. Over hundreds of thousands of years, we've selectively bred wild boars into lovely, cuddly, bacon-producing pigs. Mwah. Those are all GMOs, by the way. Nectarines are someone took plums and peaches and was like, I want to see you have sex together. And genetically combined plums and peaches, or so bred them, and produced nectarines, which folks prefer to peaches because they don't have that fuzz. I like nectarines better. I like nectarines because they are a GMO, because I think that's cool. We can also directly genetically modify foods. We have the technology to change the DNA of plant embryos and of animal embryos, and then implant them. So we can take an embryo, change its DNA, or change an egg or a sperm and change its DNA to then grow different rice or different plants or even different animals. That's how we can clone animals. We can clone, one long time ago, before you guys were born, the Dolly the sheep made huge news because she was the first large organism that was cloned, a mammal that was cloned. And basically what happens is someone took the nucleus of one of Dolly's cells. Remember, your nucleus contains all your genetic material, all your DNA. Took an embryo out of a sheep, scooped out the nucleus out of that freshly fertilized embryo, and put the DNA, it's a lot more complicated than this, and put the DNA that they took from Dolly into that embryo, put it back in the uterus of the sheep, and that embryo divided, but now it had all the instructions for Dolly. It was, and the sheep, that subsequent sheep that was born, was a clone of Dolly. Amazing also brings up some, as I was saying those things, you probably were going, ugh, because of some moral issues. Cloning humans, obviously forbidden, verboten, for good reasons. All of that stuff sounds, right? So these are amazing things. So recombinant in DNA, we can cure a lot of diseases. So recombinant DNA is when you take DNA from one organism and put it into another. So for example, we can take DNA, functional DNA from a human, insert it into bacteria to make proteins. So we can take a gene of a functional protein. So let's say insulin, we insert it into a bacteria, grow that bacteria in a medium, and we will produce insulin this way. And then we can give that insulin to inject to people who have malfunctioning insulin. We're not changing the people to make correct insulin. We're using bacteria to grow insulin for them. They still have to inject themselves with insulin. It's recombinant DNA. Gene therapy is where we do change somebody's DNA. For example, my friend Dan had leukemia. He had cancer in his bone marrow. And what happened is a person donated her bone marrow. And so what the scientists did is they killed all of his bone marrow with radiation. All the blood-producing cells in his body were murdered by radiation because they were making cancerous. They were making cancer. They were producing too many, basically. And so then once they were sure all of his blood his bone marrow couldn't make blood, they implanted this bone marrow from a donor. And now he makes that person's blood. So actually, if he took one of those, if he got blood drawn and took a DNA test, the DNA test would show him as being somebody else because all his blood cells are actually from another person. They have the DNA of that other person. The rest of his cells are all his, but his blood cells specifically, because your blood cells are made in the bone marrow, are someone else's. So if he murdered somebody and got cut and left the blood, this nice lady, I think she lives in Edmonton, would be accused of the crime. Of course, all of this is very well recorded, so that doesn't happen. GMOs, so we can, when I was young, world hunger was a huge problem in the developing world. Hundreds of thousands of people were going hungry because plants wouldn't grow in areas of Africa and Ethiopia, and the ones that did grow did not provide the nutrition that people needed. Golden rice and some other like hardier corns and things, have genetically modified foods, have put an end to that. There are business practices that these companies do that are made, that again, ethics issues start to come up, but the technology is sound. These foods have, we don't hear about famines like that anymore, where hundreds of thousands of people are suffering from famine. The famines that we have now are caused by civil war and refugees and that kind of displacement, but food is readily available. The food can be grown in these areas and is readily available and is nutritious thanks to genetic modifications, which is amazing, which is an incredible thing. And for fun things, we have tomatoes now. For a while, their tomatoes were tasting really gross because tomatoes are grown in if they're not grown in a greenhouse, they're grown somewhere far away. So they have to be they were selectively bred so that they wouldn't ripen very quickly. And as a result, the tomatoes you used to get were flavorless, mushy tomatoes. You still get those, but someone was like, well, let's figure out what proteins, what things give tomatoes their flavor, and then engineer a tomato that can survive not being ripe or traveling for a long time and produce the proteins and the acids and things that need to have it lots of flavor. And so tomatoes, as a result, last longer and taste better now. I'm sensitive to tomatoes. I love tomatoes, but I can't eat too many of them. I get like a canker sores from tomatoes. Maybe someone can genetically modify tomatoes for me. And last but not least, it's not in the books or anything like that. We've developed an amazing technology called CRISPR-Cas9. Canada has some connection to it, from what I understand, but it came around in 2014. Its possibilities still have not been fully explored, but essentially, if you wanted to change a piece of DNA before CRISPR-Cas9, it was something like $5,000, and I think it was like 20% effective or something like that. It wasn't very effective. So you'd have to do all these samples. You'd have to do this huge thing. You do all these samples, change the DNA, and all these things cost a lot of money, and then test each one, and 20% of those, maybe 20% of those, you would have the change of DNA that you wanted. CRISPR-Cas9 costs $5, and it is 80% effective. Essentially, what it is, CRISPR-Cas9 is it's part of the immune system of bacteria. And what bacteria do is, sort of as they get infected by a virus, they will cut those pieces of virus up, remember those pieces of the virus, stitch them into what's called a guide RNA, and then next time that virus injects its DNA or RNA into the cell, there's a matching piece of it already there, and what happens is the guide RNA matches up, and it's attached to this Cas9 complex or protein, and it starts to just where you insert that guide RNA, it will chop out that RNA. So basically, if a virus injects its RNA into a bacteria, the CRISPR-Cas9, because of that guide RNA, will chop up that viral RNA, and then the viral RNA can't reproduce, it can't do its thing, and the bacteria is saved. So now someone was like, well, that guide RNA is very specific. You can choose very specific pieces of DNA or RNA to cut up. What if we manipulate that guide RNA? And that's what we can do now. So we can choose a specific sequence of DNA, send in this guide RNA along with Cas9, and Cas9 will chop out those sequences, and you can cut out a piece of DNA. And then your body actually would come along and fix that piece of DNA. So if there's the cell's own machinery will stitch in a new DNA sequence. So if there's a broken, if you know you've acquired a mutation, a cancer mutation, a broken proto-oncogene, you can send in Cas9 to cut out that broken gene, and your body might fix it with a proper gene. There are an endless amount of possibilities for CRISPR-Cas9, some of them totally scary. New Zealand has this idea to use CRISPR-Cas9 to cause a mutation, a germline mutation. So that will be sex cells. So the progeny of when you change those sex cells, all the progeny will have this mutation. And the mutation they want is on kitty cats. So cats are it's kitty cats. So cats are a big problem in New Zealand. They are not indigenous to New Zealand. And so the cats kill a lot of the snakes, a lot of the local fauna that lives there, the snakes and the birds especially, a lot of birds. Cats love to kill birds. So the idea is New Zealand wants to introduce a germline mutation into cats that makes them, because there's a lot of wild cats in New Zealand, that would make them all their offspring be either all male or all female. I can't remember. It's one or the other. And obviously what that would do is then the next generation wouldn't happen, because you need boys and girls to make a new generation. So the cat's population would dwindle. At least the wild cat population, the feral cats. You can see some problems, scary problems with this. What if this technology jumps to humans, or jumps to another animal, or jumps off New Zealand and cats go extinct all over the world? What are you going to cuddle with? Don't kill the kitty cats, New Zealand. So obviously some things need to be worked out. But the future is bright. I think in terms of this biotechnology, every new technology, there's always people that are like, oh, it's going to be the end of the world. It's going to be, people usually want to do good. There's a minority of psychopaths in the world that want to do bad. And most of them don't have access to, don't have the patience to learn about these things. And we're experiencing some amazing DNA technology now. These mRNA vaccines, which I got one today. I got the Moderna mRNA vaccine today, is an mRNA vaccine. You know what mRNA is now. So I've been injected with mRNA, a gene for the spike protein of coronavirus. So I'm not making coronavirus, but now my cells will take that mRNA and translate it into an amino acid chain that coronavirus makes, the spike protein. And then my body the cells, I'm sure there's more to this vaccine than just the mRNA. My cells will excrete this spike protein, and my immune system will, I'm not sure of the details of Moderna and Pfizer guard their secrets very well. I tried to ask the person today who gave me the injection, but I don't think they did very well in Anatomy and Physiology 12 when they took it. They were very good at their job. They were excellent at their job. But I tried to have a conversation with them about it, and they didn't know some basic things. So I was like, oh, they just gave me a very standard, we need to take the vaccine and stuff. No, I know all this stuff. I know how vaccines work. Anyways, it doesn't matter. So my body will secrete this spike protein, and my immune system will learn the shape of this spike protein, and then next time coronavirus comes in, we'll be able to, my immune system will recognize the spike protein, and the coronavirus won't even get a chance to infect my cells because the immune system will get rid of it. We'll talk about that more when we do immunity. But what's amazing is that in the past, mRNA, we didn't know how to stabilize mRNA and keep mRNA around long enough where it could be useful like this, where we could make a vaccine. Now all these vaccines can be, we don't have to, because what we did in the past is we had to have something else make the spike protein for us, so make recombinant DNA like a bacteria, like another virus, make that spike protein and inject that. It just added lots of complication and expense. Now it actually is it will be a lot easier. It's very easy to make mRNA, and now we can keep that mRNA around for a very long time. So amazing things will be done now with this mRNA, because you can make your cells make things that you want them to make now. So if you need insulin, instead forget about making this bacteria make insulin or pig insulin or whatever insulin, you just inject yourself with the mRNA for insulin and you can start making it. I'm sure it's more complicated than that. But anyways, there you go. So I leave you on a good thing. The biotechnology, we are coming around a corner right now of some amazing things that are going to happen. You, my friends, are probably going to get to live a lot longer as a result than me. But that's sad too. Well, that's good. It's good for you.